Seeds are a pretty remarkable life stage in plants. And here's an example of big piles of pine seeds. And while seeds look pretty tough and inert, I think it's important to start with the realization that seeds are alive. There's living tissue in seeds, and though while at some stages they may be dormant and have a relatively low metabolism, it's still living tissue. The development of seeds obviously starts with fertilization, where an egg cell gets fertilized by a sperm cell and additional polar bodies get fertilized by other sperm cells to form endosperm. And at that point, after fertilization, the embryo develops. And this is a nice series of micrographs of the development of an embryo. One question to ask yourself is this embryo a monocot or a dicot? There are two cotyledons here, so this is a dicot seed. So as you can imagine, as this process occurs, as the embryo develops, the mass of the seed is increasing. And generally, seed maturity is thought to occur with the achievement of maximum mass. So that's sort of when the embryo is fully developed, as in this image here. That's the point of maximum mass. So if you were monitoring a seed crop, for instance, you could sample the seeds over time and determine when that seed has reached maturity. Now, in dicot angiosperms, for instance, trees that produce fruits, the stage of maturity is pretty apparent to the naked eye because those fruits are usually a great visual indication when they're ripe. That generally somewhat coincides with maturity of the seeds. But other plants, so for instance pines, it's a little less obvious when exactly seeds are fully mature and ready to germinate. Now, of course, naturally on the tree, pine cones and other cones on gymnosperms will generally eventually open on the tree and disperse the seeds at some point after they're mature. But artificial regeneration from pine cones or of pines presents some problems because it's very difficult to collect seed after it's been dispersed from the cones. The seeds are small, they're winged, and they get dispersed fairly widely. So it'd be quite difficult and expensive to collect seed from trees after the cones open and they disperse. So a lot of work has gone on in the past to determine at what stage pine cones can be picked from the tree at a point where the seed is mature, but before the pine cones have opened. So that's basically at this stage in the life cycle of the gymnosperm reproductive cycle. So recall, cones are initiated almost 2 years prior to when the cones mature. So it's really at this stage, sometime in that second summer or fall, when pine cones mature and could be removed from the tree and have seed extracted for planting or planting in a nursery to create seedlings, for instance. And this work was published by Wakely back in the 50s. So this knowledge has been around a while. And Wakely pointed out that monitoring the moisture content of cones is the proximal variable that we're interested in determining when cones are available for harvest. But Wakely pointed out that it's takes a couple of extra steps to determine moisture content. You would need to pick a cone, measure its fresh weight, and then dry it and measure its dry weight and calculate dry mass. And when in reality, he pointed out, you could actually measure specific gravity of cones, and that also is well correlated to some important points in cone development. So you can sort of think of cone development as developing along this axis, where after cones have been fertilized, so that's in that second summer, their mass increases rapidly. But during that time, when the mass of the cone, that second year cone, is developing, its specific gravity remains above about 1. But at some point, once the seeds are starting to reach maturity, basically that the cone, the woody part of that reproductive structure, starts dying, and input of water from the tree stops. So the cone begins to lose more water than its input. So that cone begins to dry out a little bit. So as those cones dry out, their specific gravity starts going down. And Wakely pointed out there was a point at about a specific gravity of about 0.86 when the cones are still closed, but the seeds have reached maturity. They've reached their maximum mass, so they are highly germinable. Whereas if those cones were allowed to naturally progress, they would continue losing mass and would eventually completely dry out, and they would open up by themselves on the tree and the seeds would disperse. So this is the point right here when trees can be harvested from the tree. They're still closed, but then they can be dried under controlled conditions, and the seed can be extracted and used for artificial regeneration. So Wakely pointed out that you can use, for instance, oil of a known specific gravity of 0.86. If you have oil with a specific gravity of 0.86, if you throw a cone in it, if its specific gravity is greater than 0.86, that cone will sink. But if that cone floats in that oil, that means its specific gravity is 0.86 or less. So floating cones in oil, you can periodically sample them, and once they start floating in oil, that's the time at which those cones are ready for harvest. Now once that 0.86 specific gravity is reached, the cones can be harvested, and these are some pictures on the left showing cone harvests. Top pictures from Rainier, and they, for most of their seed orchards, pick the cones by hand using a lift. The picture on the bottom left, that shows a tree shaker. That's actually a device that goes up, wraps around a tree, and rapidly shakes the tree. And some species, like slash or longleaf, have heavy enough cones that shaking will actually cause the cones to fall down, and they can be collected off the ground. It was pointed out to me talking with a forest manager that that manager tends to avoid the shakers because it can sometimes cause damage to the crowns and allow vectoring or input of fungal diseases, for instance, into their seed orchards. So they just pick the cones. That tractor is hauling away a big load of cones, and those cones will be taken to a drying facility. They spread the cones out on screens and allow them to continue drying as they would on the tree, but now they're drying on screens. Those cones open, the seed fall out, and then they collect those seeds and put them in a device that tumbles them and removes the wings. Most pine seeds have a little wing on them, and they de-wing those seeds, and then those seeds can then be either prepared for germination, for planting in a nursery to create seedlings, or they can be put into storage, and we'll address storage in a minute. Now, one thing about many or most forest tree seeds is that they are not immediately germinable upon dropping from the tree. That dormancy prevents the seeds from immediately germinating when they hit the soil. And that dormant phase provides a mechanism for seeds to overcome some constraint. So it may be that, for instance, dormancy enables a seed to stay on the forest floor over the winter and not germinate until the spring, for instance. Some dormancy constraints may prevent seeds from germinating under a forest canopy in the dark or under a forest canopy of the same species. Or some constraints may prevent seed from germinating until moisture conditions are sufficient to give new germinants a better chance at surviving. And we'll talk about dormancy in a second. But one thing about dormancy is that dormancy helps or enables the ability to store seed for long periods of time. So many seed, when dried below 10% moisture content, can be stored for some period of time, sometimes years and years. So one of the big elements associated with seed dormancy is low moisture content. So when seed are dried to below 10% moisture content, they can be stored. So here's an example of barley seed storage. Those are some bags of pine seed, and those seed can be stored for multiple years. So you can either build up a store of seed for large regeneration events, or seed can be stored as a genetic seed bank for future use or for genetic conservation purposes. So generally those seed are stored dried and under cool conditions, and that helps them to remain dormant and survive for long periods of time. Now for seeds of species that do have seed dormancy, those seeds must be stratified before they will germinate. And stratification, in a broad sense, just means exposing seeds to conditions which break dormancy. Now there's a number of environmental cues that can stratify seed, and it varies from species to species. Probably the most important and most common is moisture content or moisture uptake. So exposing seeds to moist conditions and allowing them to take up water. So I mentioned before that, drying seeds to below 10% moisture content keeps them dormant. And so clearly allowing seeds to take up water is generally one important step in stratification. It's so important that sometimes just the moisture uptake part is called, people call that stratification. But in reality, stratification refers more broadly to the suite of environmental cues for any particular seed that will break dormancy. But like I said, sometimes people use the term specifically to mean, putting seed, for instance, in moist peat moss or on moist paper towels to allow moisture to be taken up. They'll sometimes just call that stratification. Sometimes, in order for this to happen, scarification, which sounds similar but it's different, has to happen. Scarification just means breaking the seed coat, breaking or weakening the seed coat to allow moisture uptake. So in some species, that moisture uptake can't occur until the seed coat has been broken or thinned out. And that can be done mechanically by tumbling the seeds with an abrasive. Sometimes it's done chemically with acid, and I'll show you a picture of that in a minute. But at any rate, that would be scarification is required. And in this graph, this shows the effects on redbud, in this case, of both moisture uptake and scarification. So this is days on the x-axis versus germination percent on the y-axis. And then each line is a different treatment. So you can see in this one, they soaked the seeds in sulfuric acid for 60 minutes. That's a scarification treatment. Here they scarified for 10 minutes. That's probably some kind of mechanical scarification. And here they say they stratified. And that's a case where I said they're referring to a moisture uptake treatment as stratification. So here they just stratified for 30 days at 50 degrees Fahrenheit. You can see in all of those they have fairly low germination success. But when they combine either the mechanical or chemical scarification with that moisture uptake treatment, they get much better germination success. So redbud there is an example of a species in which both scarification and moisture uptake is necessary for breaking of dormancy and germination. So often there's some temperature regime that is necessary to break dormancy. So maybe some minimum number of chilling hours may be required, or chilling plus warming, or some other temperature regime is required. And you can imagine that, for instance, both of those, either some accumulation of some minimum number of chilling hours or a chilling period followed by a warming period, those both simulate passing through the winter into spring. Okay, so temperature sometimes is required, and then light. So we talked about either exposure to light or some specific light quality, and I'll remind you of that and show you that alder table again. So those are probably the most common regimes necessary for stratification of seed, and often species will require more than one of these in order to germinate. So here's an example of pine seed in a plastic bag. You can see there's moisture in that plastic bag. They probably have some peat moss in there, and they've moistened it a little bit, and then they're going to put it in the refrigerator and stratify it for 30 days or something like that, just in a cooler. And then at that point, they can take the seed out and germinate them. So that loblolly pine generally requires stratification. Here's an example of a scarification treatment. There's a big vat of acid and a bin that they place seed in, and they dip the bin into the vat of acid, and that thins the seed coat and will enable moisture uptake. So, obviously there's precautions required with acid treatments, but that's one way to do it. And then just a reminder, we already saw these data in the light quality lecture, but just a reminder that in this case, this is alder, and alder required, both exposure to light in order to germinate, but it also did not fully germinate if it was underneath an alder stand due to the light quality underneath that alder stand. So in this case, that alder required particular light environment in order to break dormancy and germinate. Great source of information for understanding the germination requirements of different species are these documents. The left is an ag handbook published back in the 70s by the USDA, and this is available online as a PDF. Has abundant information in it. It was updated and reprinted in a commercial form there on the right. The one on the left, that Ag Handbook, actually has some additional information. It's probably twice as long, has lots of information, general information about seed biology and so on. But at any rate, these are both really valuable documents for understanding, for instance, germination requirements. So for instance, here's a table that shows germination characteristics of different genera. So for instance, it shows this particular genus, Bellaperoni, in the Acanthaceae, is germinable. Okay, so there's a genus that doesn't have seed dormancy. When the seed are mature, you can plant them and they will germinate. But for instance, here's a genus Anacardium that requires slow moisture imbibition. So that requires moisture uptake. Here's one that requires pre-chilling and scarification. Here's another that's germinable. Here, this one, they say light ? So this may require a light treatment along with pre-chilling. Scarification. So this is a summary table, but if they also have a page or a couple of pages for each genus that describes in detail what is required in terms of treatments to overcome dormancy for that particular species. So really a valuable set of documents for understanding germination requirements of different species.